So where are our redox reactions? Derek. Perfect. Oh, no. There we go. Okay. Get rid of that. Where else? You can just shout them out. I'm not looking, but you can just shout them out. That's one. Aconitate. Or no, sorry, alpha-ketoglutarate. Yeah. Just flows off the tongue, right? Alpha-ketoglutarate. Okay. There's two. Immediately after that, alpha-ketoglutarate to succinyl-CoA. Succinate to fumarate. Oh, you were so close. That was great. Oxaloacetate. Beautiful. All right. What are the key features again for dehydrogenase? Or sorry, that gave away one of the answers. Dehydrogenase. Note that there's a dehydrogenase enzyme. I'm pretty sure every single one of these steps. The pyruvate to acetyl-CoA doesn't necessarily list one there, but isocitrate to alpha-ketoglutarate, isocitrate dehydrogenase, alpha-ketoglutarate to succinate, or succinyl-CoA, alpha-ketoglutarate dehydrogenase, so on and so forth. What else is a key feature of all these? Good. That's the key one. FADH or FAD being reduced to FADH2, NAD plus being reduced to NADH. Okay. And what happens to those eventually? What happens after this step? Yeah, dump those into the electron transport chain, and we'll come to that in a minute. Okay, so those are our redox reactions. For those of you keeping score, at the end of Krebs, including glycolysis, so glycolysis we get 2 NADH. Here we get another 4 times 2, because there's 2 pyruvates that we produce per glucose molecule, so that's another 8 NADH, and 2 FADH2. Okay, I'm not necessarily asking you to memorize those, though usually people come to memorize them over time. Oxidative phosphorylations. Very good. There isn't one. What kind of phosphorylation is here? Good. Substrate level, where we're just simply, again, the key feature of these substrate levels is transfer from substrate to another one to generate 2 new products. So succinyl-CoA to succinate gives a phosphate to GTP or ATP, GDP or ADP to generate our GTP or ATP. So 2 more ATP out of this step as well. One of the things that I often try to think about is if we were to describe describe the Krebs cycle in terms of what's the major storyline here, how would you describe that? What's the general trend or the most apparent trend in this cycle? Claire?
Reducing the electron carriers?
I think that's probably the most important one. It is chock full of redox reactions. And that then drives the electron transport chain later on. So I think that's probably the key point. Any questions on the Krebs cycle? Okay, that's all you need to know about it. That's all we're going to cover it. We'll come back to one more point with it a little bit later. I put this up here in case you want to look at it, but we've just kind of covered this, so I'll just annotate what we've done so far. 2 net ATP in glycolysis, this intermediate step of pyruvate to acetyl-CoA, 2 more in the Krebs for a total of 4. 2 2 here, 6 and 2 for a combined of 8 for 10, 0, 0, 2. So those are the numbers that we've just put together. Okay. So in this last step, then, in electron transport chain, we have oxidative phosphorylation. The name implies exactly what's happening. Basically, we have upstream of this are a number of redox reactions, oxidation of carbon in particular, to harvest high energy electrons that are going to be used to drive that electron transport chain. Where does this occur in a bacterial cell? It's in the inner membrane. One of the things that I usually do is I think about analogies here. So if you've heard of the endosymbiotic theory, for example, it says that gram-negative bacteria gave rise to mitochondria, and some of the photosynthesizing bacteria gave rise to chloroplasts. And there's some similarity in the structures, actually, and it serves as a really nice analogy to know where these events take place in bacterial cells. So, for example, here is your classic mitochondria. Here is a gram-negative bacterial cell. And then there's a cell wall in between those two membranes as well. Recall that in mitochondria, glycolysis obviously does not occur in mitochondria in eukaryotes, but it does in prokaryotes. In prokaryotes and eukaryotes, it occurs in the cytoplasm. In eukaryotes, what eventually has to happen is that pyruvate has to be shuttled into the matrix of the mitochondria, and this actually burns a couple of ATP. Just because of that process right there, eukaryotes get a little bit less ATP in the end overall than do prokaryotes. So that occurs, Krebs cycle occurs within this matrix. Of course, in our gram-negatives, occurs in the cytoplasm. And then across this membrane system here is where we will find our electron transport chain. Where do we find lower pH? So in other words, where do we find the higher concentration of hydrogen ions in the mitochondria? Is it here or is it here? The second one, the intermembrane space, right? So hydrogen ions are going to be greater in the intermembrane space than they are within the matrix. By analogy, we find the same thing to be true in prokaryotes. That the highest concentration is actually in gram-negatives in this periplasmic space. In gram-positives, obviously, they don't have a periplasmic space in the same way. That's between two membranes. So what we usually think is that the cell wall slows down the diffusion of those hydrogen ions, thereby retaining them so that they can still be used to produce high amounts of ATP through the electron transport chain. Okay. 4 major complexes. And what we're going to try to do today is to really distinguish, probably one of the key features today is distinguish, 1, what happens during respiration, and then 2, what is the difference between aerobic and anaerobic respiration. In total, by the end of Friday, we'll have talked about 3 different major pathways for metabolism: aerobic respiration, anaerobic respiration, and fermentation. One of the key essential differences between either of the respiration pathways and fermentation is the presence of the electron transport chain and the Krebs cycle. Those are mechanistically linked. In other words, if Krebs cycle shuts down, so does the electron transport chain. If the electron transport chain shuts down, so does the Krebs cycle. And it's because the electron transport chain is the part of metabolism where those high energy electrons from NADH and FADH2, that's where they get oxidized again. They get oxidized back into NAD and FAD. Those electrons will then flow through that electron transport chain and ultimately end up on what we call a final electron acceptor. Okay. Oh no, I didn't want to do that. Sorry. Well, now this is just bad. All right, let's go back to the electron transport chain real quick. Hope nobody saw that. Don't pay attention to it. First step here. NADH donates electrons to that first complex. What kind of enzyme is that first complex? And this is why I crossed it out. It has to be a dehydrogenase. It's actually NADH dehydrogenase. By getting rid of those electrons and the hydrogens, stripping them away from the NAD, now this allows for those electrons to jump right into that electron transport chain. What happens to the hydrogens? Or what could, I guess, maybe is another way to ask this. So those hydrogens that come off of NADH, where do they go? And I know you guys know this. Many of you explained this fairly well last night. Through the membrane, through protein channels to where? Where again is this outer area? What location of the cell is that? What's that? Yeah, that would be one way to describe it. So it's that periplasmic space between the inner and outer membranes. Okay, and that works just well. Okay, so those hydrogen ions are going to be moved from inside the cytoplasm to outside in that periplasmic space. These electrons then go through our series of complexes, 2, 3, 4, to ultimately end up on our final electron center. I'm not asking you to remember all of these complexes by any means, but I do want you to remember a couple of trends about them. The first one is that from complex 1 to complex 2, those electrons have to move, right? And when they move, they also lose a little bit of energy. That energy is used to drive that hydrogen ion gradient. Complex 2 is more electronegative than complex 1. What the hell does that mean? And this goes back to, gosh, first lesson, Friday. That is true. So FADH2, the electrons are a little bit lower energy. They overall won't help us to produce, they don't produce as much ATP, right? That is absolutely true. That doesn't exactly answer what do we mean by the first one is more electronegative than the next one, though. What does that actually mean? Anybody remember the concept of electronegativity? Yeah, or to pull electrons, right? And so what that allows for, each complex, 3 is more electronegative than 2. 4 is more electronegative than 3. What this allows for is essentially a pulling of electrons through that electron transport chain. And guess what's most electronegative? Actually, no. That was a good guess, though. It's not part of the actual chain itself. Where do those electrons ultimately end up, though? Oxygen. Oxygen is the most electronegative out of all of those there. So in other words, that final electron acceptor, regardless of what it is, has to be the most electronegative molecule of everything that's being used in this particular chain here. Okay? One of the things that makes aerobic respiration, I'll come back to that in a couple of minutes. I'll just stop there real quick. Okay, so we've got electrons being dumped into the electron transport chain. Ultimately, typical rule of thumb is something like 2.5 ATP are produced from every NADH. 1.5 from every FADH2. You don't need to know those numbers. But for those of you that maybe someday end up on Jeopardy or end up at a bar at a trivia contest, useful, useless trivia. One other thing that is inherent in these complexes, they have a cofactor. Iron is in every single one of these, helping to undergo redox reactions. And I know, Heather, I think you drew a picture. I can remember because I looked at it just this morning. And you wrote between each of these complexes is essentially a redox reaction, right? As those electrons are being handed from one to another, so as it goes from complex 1, for example, to ubiquinone right here, the first complex undergoes an oxidation, ubiquinone is reduced. And then as those electrons are handed off to the next, ubiquinone undergoes an oxidation, that third complex is reduced, and so on and so forth. And so what we have are just a series here of redox reactions occurring. Obviously, this generates the energy, the movement of those hydrogen ions. What type of transport? Active, passive, facilitated? Has to be active. Has to be active. And there's a couple of things that we can use as our guides here, if you'll remember from our conversations earlier last week. 1, the electrons provide the energy. 2, what's true about the direction in which those hydrogen ions are moving? Going against a gradient. They're going from low to high concentrations of hydrogen ions. And that allows for then this flow through the ATP synthase to be what kind of transport? Facilitated or passive at this point. And what do those hydrogen ions do? Ashley?
They
Perfect. Perfect. So it drives this little motor here. Oh.
What's that like channel called?
Hydrogen ion channel. ATP synthase thingy-majigger channel. I have no clue. It's a great question. I'm sure it's been named. Yeah, I have no clue. But that's going to allow for that. That basically, some people describe this inner piece as a turbine that drives then the bringing together of ATP or ADP and an inorganic phosphate here to create ATP. And this is our oxidative phosphorylation then. That's the last step. Any questions on that broad overview? And the responses that I saw, there were some really good answers that kind of hit. And what distinguished, in my mind, a really good answer from ones that were good or fair, in part was the terminology. Using terms like oxidative phosphorylation, redox, dehydrogenase. Some of those terms really help to kind of boost a response and make it very clear what you're talking about. And so I just wanted to kind of point that out. The clarity is better with the terminology being used. So this obviously is an example then of aerobic respiration. Aerobic implying O2 is being used as a final electron acceptor. How does this differ from anaerobic respiration? What's that, Kaylin? That's the big one, right? And what's used in cases of anaerobic respiration? Bree? Carbonate actually can be used. Carbonate can be used. Nitrate can be used, as Brendan pointed out. And what's the last one that's common? Sulfate's another common one. Okay? So the big thing that's different between aerobic and anaerobic is what is the final electron acceptor? sulfate, nitrate, and carbonate. I actually have these in order of which of them is most electronegative and therefore which yields the most energy. So nitrate is most similar to oxygen in terms of its electronegativity. Therefore, it yields the most ATP out of these three here, but it doesn't actually provide as much as oxygen. So one key distinction with anaerobic respiration is the amount of ATP that's yielded per molecule of glucose is actually a little bit less. And how much less is dependent on which of the final electron acceptors in this list are used. Organisms that we define as being anaerobic may use one of these final electron acceptors, may use a couple of these, but rarely do we find that they use all three. Okay. And the quote-unquote choice of which one they choose to use actually depends on the genes or the proteins that are found within these bacterial cells. So for example, there is an enzyme that's known as nitrate reductase. It actually, I'm going to go back a couple of slides real quick. It actually replaces the use of cytochrome C and this cytochrome A complex here, and actually can accept electrons directly from ubiquinone, and as the name implies, reduces or reductases nitrate to nitrite. So it donates electrons to nitrate to produce nitrite. That nitrate is the final electron acceptor. But because there aren't as many complexes in this pathway, what that leads to is a lower overall production of hydrogen ions across the membrane and less ATP being produced then by that passive diffusion. Same is true for sulfate reductase, and I don't know what the carbonate one is. I'm going to assume it's carbonate reductase, but okay. Let me show you a video real quick. And so the weekend before Labor Day weekend, so that was like after our first 3 days of class. I did not take this video, but I found this and I thought that this was a great example of what I wanted to show. Okay. So this is Lake Michigan, and I saw this at Kohler Andre, which is near Sheboygan. So this is sand, obviously, and that's the lake rushing in on it. Okay, I'll play it one more time. Pay attention to the colors of the sand real quick. Oops, I won't play it. I guess that's the only big way. Okay. What two colors are there? Black sand and regular sand. Why is the sand black? What was that?
Sulfate.
Ah, what does it remind you of that we've done? Sim test, yeah. So what's actually happening there, and if you pick up that sand and you take a big whiff of it, it smells like eggs. Okay? Why would you do that? I don't know. But you can smell it if you really want to. So, what's happening there is we've got sulfate-reducing bacteria. Bacteria that use sulfate as a final electron acceptor, reduce it to hydrogen sulfide, and that hydrogen sulfide reacts with the iron in the sand to produce iron sulfide, that black color, that same black color that we see in the SIM test. Okay? that sand can get bleached out, but usually if you dig deeper down, the sand actually becomes a darker shade of black. Because what's true about below that surface? What are the oxygen conditions like? It's anaerobic. So in other words, that sand and the water that's there is selecting, as you dig down, is selecting for microbes that can't live in the presence of oxygen, ones that must live in the absence of oxygen or must perform anaerobic respiration in order to produce energy. Okay. So I just wanted to use that as a visual to describe that process. All right. Let me just see if there's anything else that I really wanted to do. ATP. Okay. Those are the basics. What I wanted to do now was to practice a little bit with some of this. Okay? So let's start with this. I'm going to start with an example that Abby and I were talking about yesterday. It's one of the review questions. So rotenone is a drug that inhibits NADH dehydrogenase. It inhibits its ability to perform redox reactions. The question to you is, what effect does this have on the electron transport chain, and what effect does this have on production of ATP? If you can answer those two, then what effect does it have on what we would call metabolic flux? Does it change the metabolism of the organism in some way? Okay. So those are the three questions I want you to start thinking about. And I'm going to put up the image of this here so that you can refer to it. And what I'd be looking for in a response here is going just beyond saying it has an effect in the direction of the effect. It inhibits or it activates it. But saying why. So the justification part of why does that then increase or decrease the amount of ATP that is produced. Okay. So I will, I'll try to write down the question as well, but rotenone. And it's in the review questions. I'll try to pull it up. All right, go ahead. Apparently that's tilted. So hops contain what's known as isohumolones. Isohumolone. The isohumolone is a hydrogen ionophore. Ionophore. What ionophores actually do is they take hydrogen from here and they carry the hydrogen across the membrane. Okay? So what I want you to tell me is, how do hops actually block bacterial growth? What does it do to ATP synthase? What does it do to the electron transport chain? So basically the same set of questions we asked about rotenone. The mechanism is different, okay, but the end result is very similar. All right, I'll give you guys like 2 minutes because I didn't realize what time it was. I'll give you 2 minutes.